Assalamualaikum warahmatullahi wabarakatuh and a very good day to everyone. My name is Muhammad Tarimizi bin Muhammad Noor. So today I will perform a proposal presentation and my supervisor is Dr. Wan Oparahim binti Wan Osman. So the topic will be cytotoxicity effect and morphological changes of methanolic extract of Chrysanthemum indicum on HL60 cancer cell line. This is the content for this presentation. And then for the introduction, as we all know that leukemia is a type of blood cancer. And then AML is a type of leukemia alongside with ALL. And AML is signifies as the presence of more than 20% of blood cells in the bone marrow. And then APL is a subclasses of AML that manifests the translocation between two genes on chromosome 15 and chromosome 17. And in this study, we will use the cryopreserved HL60 cell line that was established from the 36 years old female that suffered from APL. So the Chrysanthemum indicum is also one of the famous herbal medicinal plants that enrich with various secondary metabolites. And the metabolites are the phytochemical constituents that are crucial in the study of pharmacological effect of the medicinal plants. For example, Chrysanthemum has shown its anti-cancer activity on human prostate cancer, DU145 cells. For the problem statement, it has been found that the uses of arsenic in ETO treatment, arsenic trioxide treatment, can increase the risk of getting liver injury. Besides that, chemotherapy also has its side effect. So that's why many researchers has turned their channel into the natural herbal treatment. So they are looking for the important and significant secondary metabolites such as phenolics, flavonoids, and so on. So for the hypothesis, there are two. There is for the alternative, there is a cytotoxicity activity of flower and bud extract of Chrysanthemum indicum on HL60 cancer cell line. For the null hypothesis, there is no cytotoxicity activity of flower and bud extract of Chrysanthemum indicum on HL60 cell line. There are two objectives. First one is general objective. We are going to determine the cytotoxicity activity of Chrysanthemum indicum on acute promyelocytic leukemia using MTTSA. For the specific objective, firstly, we are going to extract flower and bud of Chrysanthemum indicum by using methanol extraction. We are also going to observe the cytotoxicity activity of bud and flower of Chrysanthemum indicum at different incubation of time. Besides that, we are going to determine the effect of flower and bud on the morphology of the cells, and we are going to determine the apoptotic induction of the plants toward the HL60 cell line using a specific kits. For the literature review, as I mentioned before, AML is a type of blood cancer that originated from bone marrow, and FAB Association classified APL as in M3 because of its cytological characterization by dominance of promyelocyte cells. And the causes of this disease actually commonly can be found because of the translocation between chromosome 17 and chromosome 15. And then until now, available treatment include chemotherapy, arsenic trioxide, and also all trans ethanoic acid, ATRA. And this is the table that's representing. You can see that leukemia is one of the common cancer that can be found in Malaysia, represented by 3.7 cases that has been reported. And for the pathogenesis of APL, as I mentioned before, the translocation between two genes, which is PML, that is located in chromosome 15, and RARA, located in chromosome 17, was translocated, and this translocation will produce the fusion that is called PML-RA, and this will cause the productions of the abnormal production of APL, and the abnormal production of APL will causes the reducing of spaces for the normal cells to grow in bone marrow. So for the Chrysanthemum indicum, actually Chrysanthemum is a genus that has around 40 species around the world, and we can found this type of plants alongside with the East Asia region, including Southeast Asia. And its biological properties, such as inducing the apoptosis by suppression of constitutive STAT3 activation in human prostate cancer. Besides that, it also trigger the inhibition of neoplastic transformation by blocking activation of MPAK stimulation pathway and stimulation in cancer. It also inhibit the APE1 and can enhance cellular sensitivity to the alkylating agent such as methylmethanosulfonate. These are the phytochemical properties or constituents that can be found in this plant. For example, flavonoid, pyrethroid, and also phenolics. And each of these will have the significant effect of many activities such as anti-cancer agent, antibacterial, antifungal, and so on. So methodology, the research design that will be used in this study is the experimental. The flow of the study is just like in this flow, from drying, extraction, culturing, MTT assay, apoptosis assay, and also data analysis. So the extraction will be start with the drying of the samples, and then we are going to add the solvent, and then removal of solvent by rotary vapor, and then we are going to centrifuge the crude extract to remove the impurities, so that we can freeze dry the extract and then can keep the extract in the refrigerator for further use. For the culture and MTT assay, we will use RPMI or medium, and cell seeding only will be done prior to the confluence of cells, which is around 80%. And for the MTT assay, we need to prepare the solution before reading the MTT at 570 nanometers. Apoptosis assay will be done by collecting the cells and then stain with the annexin V FITC and allowed to incubate at room temperature before reading by flow cytometry analysis and fluorescent microscope within 1 hour. So for the statistical analysis, the analysis will be performed by using IBM SPSS statistic and also Microsoft Excel 2010. One-way ANOVA will be used to compare the differences of cytotoxicity from different incubation phase on bud and flower. We are going to expect there are significant activity, cytotoxicity of flower extract and bud on the HL60 cell line. Therefore, this study also attempts to explore immense scope for researchers to engage in validation of traditional claims and bioactives of this plant-related anti-cancer potential. And this study also provides knowledge for an area of advanced study to produce a new option of treatment on human acute promyelocyte leukemia. This is the Gantt chart for this study. References that has been used. I would like to express my sincere gratitude and esteem to my respected supervisor, Dr. Wan Farahim binti Wan Osman, and also special thanks to FYP coordinator, Dr. Chiu Ching Hong. Thank you so much.